Good afternoon and welcome to our webinar titled Safeguarding the World's Food Supply, Implementing a Framework for Continuous Improvements. I'm Gilad, Serial Program Coordinator and Bodybuilder at INRA Morocco, and I will be glad to host you today. Before we dive into the heart of today's topic, I would like to take a moment to thank each and every one of you for taking the time out of your busy schedule to be here. Your presence is a testament to the importance of this topic. And it's our hope that you will have this webinar with valuable insights and actionable knowledge. Today, we have a distinguished speaker, Janny van Beem, who is the Global Gene Bank Partnership Coordinator at the Crop Trust. She holds a doctorate in plant breeding and genetics from Cornell University. Former expertise includes bean and wheat breeding, gene bank quality management in liaison with National Commission for the Protection of and Equitable Sharing of Plant Genetic Resources and their traditional knowledge. She is currently involved in conservation activities with national gene banks in 30 countries. She will be sharing her experiences, strategies, and actions aimed at protecting and enhancing the world's food resources. We encourage you to participate actively, ask questions, share your thoughts, and contribute to these discussions. To ensure everyone gets the most out of this webinar, please put your full names in the participant lists and mute your microphones when not speaking. Minimize background noise. I would like to inform you that this webinar will be in 35 minutes, and after that, you will have a chance for 20 minutes for asking questions and answers. A certificate of attendance can be delivered to the participants if needed. A requesting form will be shared during this webinar. Let's make this an interactive and enjoyable experience for all. Without further ado, let's welcome our speaker, Janny van Beem, who will be discussing safeguarding the world's food supply, implementing a framework for continuous improvement. Thank you, and let's have a fantastic webinar. Please have the floor, Janny.
Thank you very much for your kind introduction. Hello, everybody. Greetings from the Crop Trust. I'd like to start out by thanking you for the invitation. This is great. I actually visited INRA in Satat not more than a month ago, and I spent a very good time there. So thank you very much for that hospitality to the Moroccan Gene Bank. It was great to see all your improvements. And so this presentation today is a little bit about that. Are you seeing right now the first slide? Has that been shared yet?
Not yet, no.
Not yet. Okay, let me go back here and share the screen with you. So,
yes, now is okay.
Okay, you should be now seeing the shell, and I will momentarily. Okay, now can you verify if you're seeing the title page?
Yes.
Okay, so there's a little bit of a lag, so I'll take into consideration there's a little bit of a lag. So the title of my presentation today is Safeguarding the World's Food Supply, Implementing a Framework for Continuous Improvement. I would also like to go over a brief introduction to the Crop Trust as it pertains to the partnership with the INRA Gene Bank. As you may know, the INRA Gene Bank has signed a couple of years ago a partnership with the Crop Trust, and I'd like to let you know a little bit more about that, what it entails, and what their work will be, the commitment to become a better gene bank to align to international standards. After that, I'd like to talk about the Gene Bank QMS. It is a quality framework that provides evidence of continuous improvement and alignment with international standards. So a word or two about the Crop Trust. It is dedicated to conserving and making crop diversity available for use globally, forever, and for the benefit of everyone. So our mandate is to provide funds for seed banks in various forms of seed banks to try to maintain those standards and even improve those standards so that they can serve their countries well. The Crop Trust is partnering with about 150 organizations in 48 countries to conserve crop diversity. It was established under international law in the year 2004, so we're exactly 20 years old this year. Our main contributors are governments, which fund us, and foundations. And the policy is under international agreement backed by about 150 countries, plus the European countries. And in this map, you can see many of the countries that are participating in some of our projects, not only partners that towards funding, but also partners that are directly involved with our activities and funding. The Crop Trust is also a partner in the management of the Svalbard Seed Vault. As you may have heard, there is a seed vault in the island of Svalbard in Norway, in the northern part in the Arctic Circle. So the Crop Trust is one of 3 management organizations that have to do with the acceptance of the seed and the conservation and the funding of the Svalbard Seed Vault. So how do we work? We receive gifts from different countries and different foundations. That is the blue square that you see here. Those funds are invested through special mechanisms, and we receive some funds from those investments, which are the green portion. And the green portion is then given to our partners through partnership agreements. For example, the BOLD project, which is the one I'm going to talk about today, that was a grant from Norway as well, and it goes straight to our 15 partners, 15 gene banks, for their improvements and their alignment with international standards. So this is the basic gift, invest, grant type of foundation that we have. A little bit more about our goals. So the donors provide a contribution to the endowment fund. Those endowment funds then go into a number of markets that we invest in, and that endowment fund is used for our primary goal, which is a long-term support and maintenance of essential seed bank operations. So this long-term, almost in perpetuity, one could say, is for seed banks that have surpassed certain indicators. For example, the number of accessions that have been well described with data, or number of accessions that have been safety duplicated. That is one of the indicators. The accessions have to be clean, phytosanitarily speaking, so that they're distributable. So we have a set of indicators, and when those indicators are surpassed, the gene bank is then a candidate to receive long-term or support in perpetuity. So we currently have 4 or 5 banks that are under this category that are receiving in perpetuity funding for their long-term maintenance from this endowment fund that you see there. Our second module that we have is for those banks that are striving to become and have not yet received or achieved the indicators. We have a second model, which is the time-bound support for upgrading and empowering seed banks. And the BOLD project is an example of this. So the current partnership that Crop Trust has with Morocco is under Goal 2, the time-bound support. It is a time-bound support for 10 years. So once those 10 years are up and the indicators have been achieved, then the banks can apply for Goal 1, which is the long-term support for gene bank operations. We also have a third goal, which is a higher political profile of crop diversity, and we have been working very hard towards this because it is not easy to make the case for crop diversity and get funding from different foundations. So this is currently what we have for our endowment fund. We are currently at 300 million. And we extract about 4% to pay for the operations that we have for different banks. So one of our major concerns is loss of diversity. This diagram is very telling. If you look at the width of each one of these colored branches that are coming out, for example, for cabbage, there used to be not more than 50, 60 years ago, 544 varieties of cabbage. This is the large green one at the left. For radishes, there was 288 varieties in a catalog that we can see as evidence of the amount of diversity that we had. For maize, 307. For lettuce, 497 varieties of lettuce. This was 50, 60, 70 years ago in the catalogs that we used to have. The bottom part of this diagram that you now see is what we currently have in diversity. So you can see that from cabbages having 544 different types of cabbages, we've gone down to 28. And from lettuces, which we had almost 500, we now have 36 known varieties of lettuce in catalogs today. So this is a huge loss of diversity, and with it comes a number of problems in terms of being able to mitigate climate change. A word about coffee, since you are in Africa, there used to be about 125 species of coffee, of which only 2 are currently grown globally. Of course, these varieties and these different land races and things are still in Ethiopia, but worldwide, I think we can agree that the loss of diversity is quite huge. This is a diagram that I like to show because it's really an eye-opener, and I think you'll understand in Morocco what this means given the 3 years of drought that you have currently have. So we're seeing that global crop yield loss is a fact now. So in the best case scenario for maize, it's a loss of 9%. For wheat, it's a loss of 7%. And for rice, it's a loss of 3%. But if we do not mitigate, meaning we go the same route as we have now, it could be a loss of nearly a third of the yield that we see in maize, about 22% in wheat, and about 11% in rice. This is a presentation from The Guardian. So by the end of this century, the worst case scenario for crop yield could be disastrous, but even in the best case scenario, it's quite highly impactful for the world's most important crops. And like I said, I think this touches a little bit the Moroccan spirit, because what I was able to see in the last 3 years from the telling of the hardships sometimes that one has to suffer from drought and heat. Myself also here in United States, the drought was terrible last year. So we can see where this is going and can readily believe it, given the drought and the heat that we have been experiencing lately. So the crop diversity is crucial for 2 reasons. The bottom ring is the obvious one, which is climate. We need climate resilient crops. We need more resource efficient crops. We need more nutrition crops. But then there's that other circle, the darker green circle on top also, that is very closely linked. And for example, on the right, more nutritious crops and more diverse cultivation really leads to a better nutrition in human beings. And also higher yielding crops and more diverse cultivation also lives or derives into better livelihoods for farmers, for example. So crop diversity is really in the immediate ring, which is the lighter colored ring around that we usually think about, but there's a wider ramification to that darker green around, which is really our nutrition and livelihood, what we are dependent upon. So while crop diversity may be becoming more important, and we are making a case for it, it is still under growing threat. And it's under growing threat in the fields, as you can see it with the farmers. It is in nature because we're losing a lot of the crop wild relatives that we have, and it is in gene banks. And that is the basic topic today is what can we do about the gene banks, how they're losing things, and how they can gain more accessions and more diversity in there. So this is where I then take it to the INRA Morocco gene bank. Specific genes are what make a crop variety resistant to heat and drought or tolerant to pests and diseases, and are therefore the basis of agricultural adaptation. This is where we go when things get serious, is what do we have? And I recall a case when I visited Morocco recently about your prickle pear and how it became extremely susceptible to cochinilla. And due to some knowledge of people there being say, we know that some of these prickle pears are resistant, they brought back these resistant varieties, and now it's being propagated. So this, we see cases around the world where the knowledge of, sometimes in somebody's head, not even in a database, somebody said, I know of 2 or 3 things that were planted 15 years ago, and we know that they were very resistant. So these things need to be documented. These things need to be noted because, there is sometimes not that much time for a turnaround. So gene banks like the one at INRA ensure that these genetic materials are safely conserved and are available for farmers and breeders to use. And I highlight farmers and breeders because we are, and I talk of myself as a gene bank, we are at the service of farmers and breeders and researchers who need this material to be able to do it, to use it properly. So a few words about the BOLD project, which is the partnership between the Crop Trust and the Morocco Gene Bank. It is derived from the Crop Wild Relative Project, which was a project before. The duration is for 10 years, so this is a long time commitment. Almost like a marriage, I would say, because 10 years is a long time, and there's always other things beyond the project that we get to do with our partners. So this is a long-term project as projects go. The total value of it was 58 million, and that is for 15 gene banks, and the donor was the Norwegian government. And you're welcome to look at the link there for more information on the BOLD project. So this is specifically the project that we are in partnership with Morocco. So more specifically, there is a work package 1 or WP1 in which we strengthen national gene banks. It's a collaborative activity between the Crop Trust and the gene bank to strengthen it. So currently we have 15 national gene banks, and the purpose is to manage, document, conserve, and safety duplicate crop diversity and make it available to Moroccan farmers and breeders. So there are heavy fighters here. Manage, first of all, the management of it is not as easy as it may seem. It needs to be maintained alive. It needs to be clean. The viability needs to be above 85%, and it needs to be categorized properly for the proper distribution, legal distribution of it, so that intellectual property rights are not sort of taken over. So manage in itself is a large word speaking about trying to align to international. Then it is to document. How do we collect the data from it so that it is actually useful? As I have said many times, the seed itself has absolutely no use by itself. The seed has value when it has data. It's the data that give the value to the seed. So to document this, these accessions is very important. Then to conserve. Many different things are conserved in different ways. So they're conserved in the field, they're conserved in vitro, they can be conserved in cryo, and of course they can be conserved in seed. So the decision needs to be made by the gene bank manager. What is the best way to conserve? And safety duplicate. This means a copy is sent to Svalbard or somewhere else of your choice, so that you have a full copy of your accessions just in case anything happens. So one of our outcomes is, of course, is to manage crop diversity for the long-term use, again intended for farmers, breeders, and researchers. And also, we feel that there should be an outcome where there is facilitated use of new crop diversity by farmers and breeders in the development of advanced lines. So there are other work packages within BOLD. This is work package 1, but work package 2, 3, 4 is helping in different forms to bring the group together and facilitate new use. So if there's new germplasm availability in other work packages of BOLD, that they would be available to the Moroccan Gene Bank and other participants so that they can promote this to farmers and breeders. So we feel that it's not just, let's just save it in a large freezer. It is much more than that. It is try to introduce material from others and also make those your accessions so that you can promote them to your farmers and breeders. And thirdly, strengthen linkages with seed system actors. So we think that technical and institutional innovations should enhance the crop diversity through the linkages, for example, with private companies, seed multipliers, and all of these other actors. It's not just the gene bank, but there are other things that come into play. And we believe that this is a very strong work package one. So it not only looks at the seed already in there, it's the actors, other diversity coming in, and then what is out there in nature that still needs to be preserved. So who are the national gene banks that are in work package one? So there are 15 of them, and it's Azerbaijan, Bhutan, Cuba, Ecuador, Egypt, Laos, Lebanon, Morocco, Pakistan, Peru, Sudan, Tanzania, Uganda, Vietnam, and Yemen. So you see that there's quite a nice scatter of different countries with different issues, different germplasm, so that altogether we have a good amount of diversity that then can be created into new accessions and new lines, new breeding lines, and then possibly shared. This is the actual group that we have. At the top right, you can see Benjamin Killian. He's our leader. And next to that is myself, and next to me is Ali, who is a very integral part of this group of 15 countries. So then moving on into implementing a framework for continuous improvement, a few words about what we're actually doing to get to where we're doing. And the Morocco Gene Bank is pioneering the implementation of a tailored quality management system. What does this mean? This means that we don't have a system that is imposed on all of our partners, because everybody has different challenges in their own country. And we feel that some things take priority over others. And in that case, when I visit or when other people visit, we try to work with the situation that we have and get as far as we can under that particular scenario. So everybody, Morocco is not the same as Cuba, as you well know. It has very different conditions. And it's not the same as Yemen currently, which is under a different condition. And then, for example, Azerbaijan. So these are very different countries with very different challenges. So that's what we call them, implementation of a tailored quality management system, because the differences are so great. So the quality management system is a tool that helps direct the gene bank activities to meet user needs and regulatory requirements. In red is perhaps the most important part, which it provides evidence of continuous improvement and efficiency and effectiveness. So many gene banks already have quality, and many of those of you watching today already have quality in your operations, but it is really difficult to see. There is no transparency. If I were to ask you in your activities, do you have a standard operating procedures? Do you have a risk management policy? Do you have data associated to what you do? And is the data aligned to international standards? Many of the answers to those questions are no. So this system, which I will speak about, is a system that is transparent. It provides evidence of continuous improvement. While you have the quality, it also allows you to show what you have and even brag about it. Say, I actually have standard operating procedures that have been written and audited and improved through time. So in other words, QMS, quality management system, provides evidence that excellence is a habit, so that it's not a really big chore or an imposition. It's the way you work. It's a modus operandi of what you do. If you want to look at it diagrammatically, you already have quality. You're already standing on this quality continuum. And what you would like to do is to take steps as best as you can take on this job, take steps towards something that you've identified, a way to go that is going to be better. So this QMS is a tool, and this is the steps that I'm showing here, that assist gene banks in assessing where they are. Are they at the very bottom and just starting? Some gene banks that I've seen that I've visited are kind of at the top. They already have procedures written. They already know what the standards are to align. So first of all, you need to determine where you are in the continuum. The second thing is you need adherence to international policy. That is the legal part. And I think that that needs to be done first is what are you aligning to? What is your expectation in terms of alignments also for international standards? Some people don't even know what the standards are. We have to identify what those standards are. And I do have a list, which I'll share with you later, of what exactly is it that we're aligning to. And obviously, when you have this adherence to international policy and alignment with international standards, you then have a higher efficiency and effectiveness in gene bank operations because they almost force your hand into being very efficient and saying this method, we're a little bit clumsy on it. Let's do the more modern method. And in that way, you're capturing some of the efficiencies and effectivenesses that are available to you there. So in the gene bank QMS, it emphasizes 8 essential areas and 8 essential SOPs that allow gene banks to plan and improve and monitor progress. So I'd like to talk about that now, and with the understanding that whomever you are, welcome breeders, welcome those that are conserving other genetic resources that are not plants, entomologists, anybody that is there. Basically, what I will be talking about applies to you. You can do exactly the same things. And I have a concluding slide at the end as what your steps would be. But this is applicable to anything you do. The CGIAR just went through a whole process management, which is basically this, of their breeding program. So the breeding program now has essential elements, which I will talk about now, and essential basic procedures. So if you see me saying gene bank QMS, it's not only the gene bank. Anything you do can be applicable to this type of management. So if there's anything you can remember about the gene bank QMS, and I'm trying to make it as simple as possible, is 8 and 8. There are 8 basic elements in the quality management, and there are 4 gene banks, 8 basic procedures. If you are in another field, your procedures, your basic procedures might be different. I would envision that for breeding, it would be slightly more than 8 for your basic procedures. In other words, to cover your entire operation, but that would need to be investigated. But to remember for now is the 8 and 8. So what is Dr. Ali Safri doing with his team in the gene bank? So to remember this, there is an octopus that I like to talk about because it has 8 legs, and we're going to talk about 8 of the important elements. So the mistake people make sometimes is saying, well, SOPs are written, therefore we have a quality management system in place. And they equate SOPs with quality management. Nothing could be further from the truth. And allow me to show you how. So the SOPs are in the first leg, which is science and operations. That is a big leg, obviously, of any endeavor, any unit, any program under, for example, INRA Morocco. The science and the operations is first. This is where the SOPs are written, but that is only one leg out of the 7. The second leg, for example, is risk management. The third leg is staff management. You need to have control of your risks, and you need to have control of your staff. So if you don't provide evidence of risk management and staff management, you really do not have a complete picture of what quality management is. The SOPs are not the only thing. The other very important part is EIR, which is equipment, infrastructure, and reagents. We find this to be a very, very important part of what we do. Many equipments are old, they're not calibrated, they're not properly maintained, including the growth chambers where things are kept. And so we find that a good amount of control over equipment, infrastructure, and reagents is a very good way. Policy is very important because it is the legal part, and we should always be within the legal realm. User satisfaction. User satisfaction is absolutely necessary. How many times have you gone and asked the farmers if your work is actually useful? That is a job of a gene bank manager. He needs to have a network of farmers for whom he's working. They have to be identified. They have to be close at heart to the gene bank and to the breeders. They have to be participating in participatory breeding or demo plots where the gene bank shows. So that user satisfaction becomes one of the most primary legs of what we do. Is, are we really doing our job well in terms of user satisfaction? User being breeders, researchers, and farmers. That is whom we need to be asking if we are doing our job. Information management, absolutely necessary. As I said, the seed itself by itself is not that valuable. Nobody's going to be planting seed that they don't know about. It would be catastrophic. We need the data, and this is where information management comes in. We need data, and we need to know what it's about. Supply chains. Supply chains is who do we receive our supplies from? They dictate our quality, and therefore supply chains are absolutely necessary. These 8 legs are supported by proper documentation for capacity development and as evidence for donors and auditors. So documentation needs to be there for transparency. The 8 basic gene bank SOPs, as I said, 8 elements. We've just spoken about the elements. These are the 8 basic SOPs of a gene bank. If you're in a different unit or a different project, they would be different. You would need to map these out yourself. But these are the 8 for for gene bank SOPs: acquisition, conservation, regeneration, characterization, distribution, safety duplication, information management, and collecting. So the minimum, most simplest bank, for example, 1 crop and 1 conservation form, only seed, will have these 8. That is the very minimum. This gets obviously a little bit more complicated when the operation is a lot larger. For example, you have seed and you have field banks and you have in vitro and you might have cryo. Already you have 4 different forms of conservation. That makes your map, your operational map, much more complex. And therefore the number of SOPs, standard operating procedures, to cover your entire operation is more. The other thing that complicates a little bit is the biological status. We have cultivated crops and we have wild crops, wild crop relatives. So if you have wild crop relatives, it has to require a vast amount of knowledge to be able to preserve them well and preserve them at a viability level that is indeed useful. So again, you will have to have, it will force your hand into opening up to more procedures. So this is a diagram of one of the works that we did, and this takes half a day to a full day to achieve. It seems so simple, little sticker notes on the board, but in actuality, it tells you the complexity of the business. Currently, INRA mapped 12 candidate SOPs, so they're not basic like 8, they're 12. It has many more SOPs that it needs to cover the entire operation. And in moving to the new bank in Rabat, by chance, I was there when we were taking a moment to talk about the new Rabat gene bank. Congratulations on that. I think that's an enormous achievement. But when Ali and the team and the gene bank move to Rabat, it will possibly grow to about 20 SOPs in total. So it is more than double, almost triple the number of what a basic gene bank would see to cover the entire operation. So this is a very important first step. This little diagram, this little exercise that takes into consideration every single activity in your program or unit. And this translates to something like this. This is, again, a very simple gene bank. As you can see, it has 9 procedures. It is Africa rice, located in Côte d'Ivoire, and it has only seed, and it only has 1 crop, rice. So you can see how complex for only one seed and only one crop. This is what it looks like. Each one of those columns is a procedure with an owner and the number of activities there in the middle that are very clearly highlighted what is included inside of these different SOPs. So Ali will have a total of 20. So his diagram will be much wider, and it will also include the totality of all the activities within that gene bank operation. So I mentioned before, and I'd like to express that now, is what exactly is this quality management aligning to? In the diagram of the stairs, of the man walking up the stairs, I said that there had to be an identification of where we are going. The number one book that we align to is the FAO Gene Bank Standards. The Gene Bank Standards have three versions: the 1994, the 2014, and the 2022. And on top of that, it has codes of conduct. For most gene banks, this would be enough. This would be a good project to take on and align to all of that FAO has given us for gene banks. That is 4 different publications. It has a lot of information. The 2014 is a book that is about 2 inches wide. And now with the practical guidelines, it's 4 separate books. So just by itself, that would be an undertaking for any gene bank to align to these FAO standards. But that's not what we are doing in BOLD at all. We're also aligning to ISO standards, particularly the last one, 20387, which is for biobanks. So if you're following the Gene Bank QMS for the BOLD project, you're also automatically becoming aligned to ISO standards. The third one that you're also aligning is international standards for phytosanitary measures. You cannot keep a proper gene bank without having some phytosanitary control that guarantees that you're not spreading diseases. So it is very important for you to be in line with the standards for the IPPC, which is International Standards for Phytosanitary Measures. There's also the International Standards for Rules Testing, which is under the International Seed Testing Association. So all the seed companies have one big manual, and it is the International Rules for Seed Testing. This is, of course, for seed companies, but we are in the business of seeds in the gene bank. Therefore, we should have some alignment with the international seed companies. This is where it comes in, the ISTA handbook. So if you are working in BOLD and in the crop trust towards a gene bank quality manual, you automatically have that also under the number of references that are there. ISBER, we also follow ISBER, Best Practices for Repositories. It is a Canadian organization, and they have very good manuals for storage, retrieval, and distribution of biological materials for research. This is a little bit bigger than just plants. It's all biological materials, but an excellent reference for us. And we continually update our versions that they produce and become aligned to them in our work. Of course, there's also international policy like Convention of Biological Diversity, international treaty. There are best practices, excellent best practices and manuals of very, very wise people. They've been written a few years ago, decades ago, but they're still very valuable, and we follow them, and it is incorporated into what we do. So as you can see, any gene bank now with this gene bank QMS needs to stay up to date and abreast of all of these documents to be able to implement them correctly in their gene bank. Not only just write SOPs, but write them in alignment with what I have just shown you. So this circle that you see here, that is the octopus that I showed. So it's science, it's policy, it's risk, it's staff, it's equipment. That is what the elements are. We're also talking about the management of the quality management, and that is improvement plans. How do you manage all of the quality management? Who is looking ahead and doing improvement plans and KPIs and continually teaching staff, sending staff to different workshops? So there has to be a large capacity building component to it. That is very important at the top of that 8 element there. And we're working on that as well. Again, I said there has to be adherence to international policy. That's the top of a pyramid. That is super important. But most of the work that we do goes right below that. And I left this slide not for you to read it now, but I think it's important. If you would like a copy, I left a copy with my host today. If you would like to see it, please ask them for it. I left that bottom of the graph because that is where most of the evidence is. If people want to see any evidence of quality management, the big red arrow, the bottom of it, is where we normally are working to produce the documentation and gain some efficiencies from using more modern methods and more technology. So this pyramid is actually the basic structure of everything we do on one slide. This was a very hard slide to get because it had to be, show everything we do in quality management and exactly what document is being produced under what category or what element. So that is what the INRA Morocco Gene Bank is undertaking now for its second year in a row. A few words about risk management. This is an important part about what we do. Currently, each bold gene bank is creating what is called a red book of risks. It contains a risk assessment table, a business continuity plan, and an emergency action plan. This is a version of what the risk assessment table looks like. So we've already identified 147 risks. And on the right-hand side of that table, you see very specific mitigating steps. We already have a copy of that, and we're trying to create an element of risk management in every gene bank manager so that he is well prepared for any risk eventuality. There is a, excuse me, a business continuity plan. These are actual pictures on the top of the Syrian gene bank, and the bottom is the Filipino gene bank. So 2 gene banks that have been destroyed by civil unrest and fire. So we do need a continuity plan should anything happen. Where does the bank go to continue its operation in spite of adverse effects? Lastly, there is a third section in the Red Book of a first responder plan, and it is the center's emergency response to fire. Usually this is only 1 or 2 pages, and usually most institutes already have a response, emergency response to fire, illness, or something like that. So that is within the Red Book of Risk. I'd like to talk a little bit about data management. Seeds by themselves, as I have said, are of little use to farmers, breeders, and climate mitigation. The value comes from adding the data. And that is one of the jobs that the BOLD project is emphasizing quite heavily, is things need to be characterized and evaluated so that it provides users with the necessary information to select genetic materials correctly. Otherwise, you're just giving people, to be honest, garbage. You're just giving stuff that you don't know anything about. They do a lot of breeding, they do a lot of planting, and then the gains in yield, the gains in abiotic and biotic resistance are very low. So we need to have data. Data adds value. And for that reason, annually BOLD partners, including the Morocco Gene Bank, are regenerating and characterizing accessions to obtain internationally standardized data and fresh seed. So this increases the value of Moroccan gene bank, Moroccan germplasm. So again, this is data, but it's not just data, it's internationally standardized. We have to standardize so that when it gets uploaded to a global platform, that it is something that other people recognize, and you're not just using data from your own memory or wisdom. These are internationally standardized data that then are shared globally, and we can learn a lot about that because we have a way to compare what yield really means, what resistance really means, what does that really mean in your mind? It's a standardized form of data. So currently in CETAT, there are fields, I think 1,300, if my mind serves me right, 1,300 accessions that are currently being grown and characterized in CETAT so that this adds value to the Moroccan germplasm. So the ultimate output of this project would be the creation of what's called a Morocco Gene Bank quality manual. It is not your normal manual. This is not a manual with simple protocols. This has all of these factors in it. Gene bank standard operating procedures. It has staff management. It has equipment, infrastructure, reagents, policies, risk management, and information management. All of that is what we call the quality manual. And it is really the best product. After 2, 3, 4 years of working on this, the Morocco Gene Bank will have a quality manual, which then is evidence of their quality management and continuous improvement. That is sort of the ultimate goal at what we're getting for. So my last slide, if you're in another business, in another project, next steps for implementing quality management or something like it. You don't have to talk about quality management. You could say continuous improvement. You could call it continuous improvement. The first thing you would need to do is discuss which of the elements, those 8 elements in the octopus and SOPs or documentation are most important for your operation. In other words, prioritize your improvements. You need to have a priority. What is the most important thing that you have that needs to be controlled, that needs to be improved? You need to identify that first. That would be your first step. The second step you would need to do is you need to map the entire operation with all your staff there. You need to say, what is our operation? How many little sticky points, sticky notes do we need to cover the entire operation, everything we do? And you need to put them up on a board and see the entire operation. The smallest operation I have seen in a gene bank is 8. The biggest operation I have seen in a gene bank is 47 different standard operating procedures. This is a complicated, very sophisticated bank, and it is completely tailored. It is your prerogative to choose what those SOPs are and to explain the complexity of your business. Thirdly, make a plan, maybe a yearly plan, including what needs to be done and who will execute the improvements. For example, do you need to write an SOP? Do you need to buy equipment? Do you need to collect fresh data? So how are you going to go about and who is going to do it? So the actual activity and the timeline needs to be there. So some form of planning, proactive plan needs to be there so that you can go up those stairs that I was showing you and do it in a way that it's not going to put too much stress because you already have a job. So it is just a way for you to be able to provide evidence of what you already have, provide some transparency. My number one advice is don't start from zero. Ask others for assistance that already have templates and already have policies written. You may have an old manual that you can use. There is always text somewhere that somebody wrote. Don't start from zero. This takes a long time if you start from zero. My advice would be look at the old manuals, talk to the elders, people that have been there for quite some time, and remember publications that are around, manuals that are around, and use those to begin your journey for continuous improvement. And I think with that, thank you very much. I'd be willing to answer any questions that you have.
Thank you very much, Jenny, for this informative presentation about gene banks and gene sources and also the quality management system. So I will see for the audience if you want to ask questions related to this topic. Before that, I will just have a little chat about this document because it's very important to use genetic resources, especially now with this climate change, for drought, for water scarcity, and to find solutions in order to overcome this phenomenon. So we have one question from Dr. Lamari from INRA Stats. Yes, Amari?
Okay, okay. Did you hear me?
Yes, we can hear you. Thank you.
I would like to thank you, Jenny, for your presentation about, gene bank and all this clarification about, the gene bank. just I would like, to ask a question about, the, if you want, the legal issues, about, the exchange of genetic material between countries, which is really a crucial question about, when it comes, to exchange, of this material between different countries, and we have different regulations from each country has its regulation, in terms of, if you want, exchanging, materials. So, any exchange within the gene banks should be, limited, to the gene banks. Gene bank from Morocco with, I don't know, the other one in Europe or in US. Or, if when it comes, to country, is it possible or not?
Yes, thank you for your question. Yeah, very, very, very good question, I might say, because one of the things that the gene bank manager has to do is to be completely knowledgeable about what the rights and regulations are regarding the distribution of plant genetic resources from that country. So the gene bank currently has 3 ways of distributing. Number 1, it can distribute internally to INRA. That is a very straightforward distribution from the gene bank to its breeders, its entomologists, or whomever else within INRA. So that is the first kind. The second kind is the distribution of the gene bank within Morocco. So the Moroccan gene bank obviously works for the Moroccan people. That is the number 1 client, right? The number 1 user. So there are exchanges of material that need to happen between the Moroccan gene bank and the Moroccan people. So that is a different type of agreement from the INRA one, the within the institute. So the Morocco one is a different type of agreement. Farmers, for example, especially smallholder farmers, have a right under many international policies to receive the seed without any agreement, because they have been working for us for 10,000 years or more, already farming. So the idea is that we give the farmer anything he wants, so long as he's using it for subsistence and he's planting and he's sharing with his neighbors, that we give for free. So that is kind of another type of distribution within Morocco that should be taken into consideration. But there might be companies in Morocco, there might be universities in Morocco that have a different type of agreement. And Ali now knows what the actual agreement is. He's undergone some training from before and since the BOLD project, where the very particular material transfer agreement is used within Morocco. And then it's the third kind that you talk, which is internationally. And this is the one where you have to be a little bit more careful, because some things may be easily shared and others may not. Some things may be of the Moroccan government or the Moroccan people, or they may be belonging to a breeder. And so at that point, it's a line in development or developing material. So it cannot be categorized the same as an accession, which is an accession is another type of categorization. So knowing the difference between the different kinds of accessions and then your users, whether it's an international user, a national user, or an institutional user, requires you to be able to use the right material transfer agreement. That is all something that the gene bank manager needs to know and be able to apply. Sometimes even a special agreement needs to be made for a special study. So then a different material transfer agreement needs to be developed. So it's not one fits all. It depends on whether you're a national, international, or institutional user and what the actual germplasm that you're distributing is being used for.
Dr. Farrahi, can have your question.
Thank you, Jilal. Thank you, Jenny. Completing the question of Dr. Ammari, this question of sharing genetic material with probably the users outside of the country, that probably causes more challenges and probably you can there is as you know, Jenny, there is the Nagoya Protocol, the access and benefit sharing that should be also taken into account. And in Morocco, we are in the process of establishing the law for the access, the Nagoya Protocol, the access and benefit sharing, and
Correct.
that's also as an important part of this sharing of genetic material with outside countries, and that's something that should be also discussed within the gene banks and agreed on at the level, at country level. That's just a point I want to mention for this also, this material transfer agreement.
Yes, absolutely in agreement with you. I mean, there are legal policies right now, legal entities that kind of dictate how we do things. And thankfully for that, because it gives you the freedom to categorize your material. So you could categorize, for example, some materials to go under the international treaty. This means that they can be transferred for only 3 purposes: breeding, research, and training. So they can be, and they cannot take and get intellectual property rights on those materials. It's very clearly written. So if you use the SMTA, which is that type of agreement under the international treaty, you would then have that agreement be the bind between you and whomever the recipient is. You also have another way of categorizing different genetic material, and that is through the Nagoya. So anything new that you are bringing in and collecting could well be under Nagoya Protocol. And in that way, you use your own MTA. You say, well, if you end up using this, we would like to be acknowledged. We would like to have royalties on this. I mean, you can stipulate exactly what you want under the categorization. That categorization, that column under the database is a very important column because it tells you which one of the treaties you're using. The treaties or conventions, right? So that categorization is absolutely important, which means the gene bank manager, which is the recipient of all these material, whether they're breeding lines, advanced lines, released varieties, or accessions that have been collected 50 years ago, either way, they have to be categorized so that you know which is the material transfer agreement to be used. So that is an important thing of the job of a gene bank manager, to properly characterize things in order to be able to use, send them to a user with a proper agreement. There are several formats of, for example, MTAs. So those that are under Nagoya Protocol. I believe I shared some with Ali of MTAs that have been used in the past, where things are different from the SMTA, which is under the international treaty. That is a different format altogether. But knowing that obviously is part of the responsibility of the Moroccan Gene Bank.
Thank you very much. So I have a question or comment about this. So I believe that in order to have at the same time conservation, regeneration and use of genetic resources, I think that it should be a multidisciplinary team in order to, for example, breeders, they can at the same time, for example, Ali do regeneration, and the breeder can identify the best material for him. Also, the pathologist can also assess the different diseases for this one. So like that, I think it could be also in order to have these 2 elements from QMS, like service and operation, and also information management at the same time. So we will have at the same time the data, and we will have regeneration, and also for the users, breeders can also have this one. Second one is that we can also have information within conservation. For example, for cereals, we can conserve spikes instead of seeds. Why spikes? Spikes, it gives many information on the accession. For example, for barley, 2-row and 6-row, from seeds, wooded. So different information that can we have it at the same time. Third one is that climate change also not only affect farmers, but also will treat gene banks, because with water scarcity, so for the regeneration, there is a big threat to have regeneration and irrigation. So this one. So I believe that in order to have like regeneration, also use, I think for benefit sharing, to have like on-farm collections. Under regeneration, we can also distribute different collections to farmers in certain areas in order to regenerate, and at the same time to have to spread this diversity, because
Exactly.
now with this the lack of diversity, so we can spread this through this kind of actions.
Yes.
Okay.
Yes, absolutely. I couldn't agree with you more. I think it needs to be a gene bank manager working alone is really a very small gene bank. You need to widen the perspective, and you need to work interdisciplinarily, like you say. So, for example, when I worked in the gene bank in SIP in Peru, we had the phytosanitary unit constantly coming to visit our fields, inspecting them, telling us what was resistant, what was susceptible, and taking notes on the degree to which it was. So that is something that needs to be done, worked very closely together with the phytosanitary unit. The gene bank needs to be working very closely with breeders in demo plots that the gene bank puts out in regeneration fields. It's very good to walk breeding lines, breeders' fields, as well as for the breeders to walk gene bank fields, because you get a very good sense of what is working and what could have more potential. How do you bring these good lines out? And lastly, the farmers. I think we're doing in Africa with another project called Seeds for Resilience, a lot of work with farmers. And it's incredible the discrepancy between what the farmer thinks is good to what the gene bank thinks it's good. It's an eye-opener. You know, when you invite farmers onto your fields, what you think is good as a gene bank manager, as a breeder, sometimes has nothing to do with the reality out there. They're looking for another growth habit. They're looking for another type of, so you need to be in contact with farmers, especially women farmers these days. Women farmers are taking a bigger role. They're the ones that take in the grain. They're the ones that are feeding their children. What is it that they need? Do they need crops that boil faster so that they consume less firewood and less electricity? What exactly does that female farmer look for? And if she's working with her husband, what are they looking for together? You know, how can we better service them? I think to be in touch with that is really an eye-opener, because they have ideas that we have no way of knowing what the challenges are or what their customs are. You know, it's about a customs. You can give them a red bean and they may like it, but if you provide them with, say, a white bean in some cultures, they absolutely love it. You know, it's a mindset. We need to take that into considerations as breeders and gene bank managers. And also, you know, how best do we give them material that is actually going to survive the season and not just die out because of heat or drought? So absolutely agree with you with that interdisciplinary team in the work that we do.
Thank you very much. So I think Dr. Amri also want to ask something. Dr. Amri? Raise his hand.
Hello.
Ahmed.
Hi, Yanni. How are you?
Hi, Ahmed.
Good. I think I'm really happy to see that, I mean, you are still continuing to support the national gene banks.
Yes.
Okay. My only concern with I'm sure that there will be a lot of more than 90 SOPs or 80 SOPs as in the gene bank of Morocco. The new gene bank will be extended to, I think, to microorganisms, to other crops, like, I mean, field gene banks, to animals. So my thinking is that with this present status,
Yes.
it will be really hard with 2 persons or 3 persons in the gene banks to handle the diversity of crops, the diversity of, I'm talking just about seeds now. In
Right.
terms of curating the diversity of crops, I think there is a lot to do.
Yes.
I think I will stop here. Just hoping that this can be taken seriously by the management in the future for the new gene banks to really have all the diversity of expertise needed. And the other thing is that I'm sure that there will be opportunities for networking to handle this at some stage. Thank you.
Yes, I agree with you, and very good to see you, Ahmed. I couldn't agree more. And I have to bring a little bit of a sensitive topic here. Not too many years ago, there were 30 people working in a gene bank, or 30 people working, at least in Setat, on many of this diversity. I think you're down to 1 or 2 people. And what I have described is a complex gene bank. National gene banks are complex because they have the mandate to curate a huge number of crops that service the whole country. And not only that, service a country that is going into climate change, which is even a bigger thing. So I absolutely agree that it would be advisable for you to have a good look at how many people there are in your gene bank currently that could cover the entire operation. 20 SOPs for the new bank was a conservative and humble estimate, because it only looked at the part of plants. If on top of that, you are doing animal conservation and you are doing microorganism conservation, this becomes an even more complex gene bank. So obviously, this cannot be handled by 1, 5, or 10 people. This has to be a crew of very well-trained people in their own disciplines that know how to conserve it best, know how to distribute it best, and have a safety duplication and also a database that is useful. So the data has to be there for all of those 3 different types. So, I mean, the amount of knowledge that there currently is dwindling, and it's only based on 1 or 2 people. Should those people decide to leave, all of that wisdom goes out the door. So this is a very precarious situation. And Amri, thank you for bringing it up. It is a sensitive topic, in every single national bank. There has been a dwindling of the number of staff, and obviously a dwindling of the knowledge and the succession. There has to be a way to bring back the knowledge and have enough people that we don't run the risk of losing 1 person and having all the knowledge lost. So I think that is a very valid point.
Thank you very much, Jeannie. Farahi, you want also to ask something?
Yes, Jilal. Dr. Jenny, I have a question for field conservation, in situ conservation. Probably Dr. Amri mentioned a little bit. Do we at crop trust level, do you have SOPs for field crop conservation, for in situ conservation? We are in this seminar. It's about ex situ gene banks, but
yes,
my
yes.
question is about in situ conservation.
Yes.
Is there any SOPs at the crop trust level?
Currently, the BOLD project is not emphasizing in situ conservation, but we do have field collections, and field collections is part of the gene bank. So if you have an olive grove, or you have cassava, or you have plantains, or you have fruit trees, for example, that would be part of what we do, actually. And we do have SOPs for that. So that can be arranged for you to see the field collections or field conservation of particular crops. That we do have. The in situ conservation, no, because it requires an expertise, and that would be sort of a second step that we would do. We need to organize our house, wash and sweep and broom everything in the house with what we have, properly conserved, properly backed up, and very well documented what we have in the house. And then once we have that in the house, both seed and field collections, then we're going out and doing the other one. There are parallel projects that work in situ and try to conserve in situ. But for us in BOLD, and particularly in the Crop Trust, our concentration is now ex situ, including field collections.
Thank you very much. Anyone who wants to ask questions about genetic resource and gene banks? Yes, Ahmed. Yes, Ahmed.
Do you hear me?
Yes.
Yes,
Hello?
we hear you.
Yeah, thank you, INRA of Morocco, and Madam Dr. Jenny. And I'm Ahmed from Ministry of Agriculture, Ahmed Al-Alwani. And I'm interested in this subject related to the genetic enhancement of plants and also of animals. But in this topic related to the safeguarding supply chain using genetic enhancement, I would like in our work in Sahara in Layun, and I'm asking how, for example, this gene bank can help us in improving productivity of corn, hydroponic corn.
Mhm.
Because we have received critics related to this corn, hydroponic corn, that in its yield, in the yield of on this on the productivity, the effect of using corn, hydroponic corn in animal production for
Right.
in that sense. And thank you.
Yeah, again, I mean, for hydroponic corn, you could have a number of characteristics that make that particular accession that you're using more well suited for that type of cultivation, right? So I think it would be a little branch of the gene bank could, for example, nominate some candidate candidate accessions that could work for hydroponic SOP. So for example, you could nominate or select some accessions that work best under that. Not all accessions will work well, but for example, from the maize diversity that you have, you could use some of those accessions. The other thing you could do is, if it's not working with the accessions you currently have, there is a huge network of people that would be willing to send you some maize seeds that could work, that they've already tested and could work under hydroponic conditions. And so not only from your bank, but also widen up your perspective a little bit. Are there other lines that could be available that you could either do breeding with or use directly, and see if those work for you? I think that that would be the question. Once you get those yields and you're happy with that, then that would be a way that you could go to develop similar breeding lines of the same families, for example, or the same accessions. You could develop some there. So I think that that is a very specialized use of the accessions, and I think it would be worthwhile looking into to see internally if you already have some lines that work under that system, or maybe even externally ask for from partners, ask for some of those lines to be sent to you, propagate them, and then use them as possible hydroponic corns.
Thank you, Jeannie. Anyone who want to have questions, please feel free.
There's a question there saying, can we have the presentation? And yes, by all means, I have shared my PDF presentation with the hosts, and you're most welcome to have it. Please share it as you will, to whomever wants. Please share it as people ask.
Thank you very much. Thank you, Jeannie. So I would like to thank the director of INRA for hosting this webinar, and also for the scientific and technical committee of the webinar series of INRA. Special thanks to Jeannie for this interesting presentation on genetic resources and gene banks and how to improve this one through QMS. So it's very interesting and very useful, especially for breeders like me, to learn that we can also be a part of this interesting framework in order to enhance the use of genetic resources, and also to help genetic gene banks in characterizing the genetic resource there. So it's very important. So thank you very much, Janie, and special audience. Thank you very much for being with us for this webinar, and see you next time for new webinar. Thank you very much.
Thank you, everybody.
Thank you very much.
Thank you. Have a good afternoon.
Thank you very much. Bye.
Bye-bye.